a video to help you through the activity on characteristics of amino acids. This activity requires that you understand a lot of the material that we've covered before. You'll want to review the information about chirality, functional groups, and conjugate acids and bases. Please click on the links below to review this information before you start. In front of you is a table of amino acids. You'll want to get very familiar with this table. The amino acids are divided into four main categories: the nonpolar amino acids, polar amino acids that are neutral, or in other words, they don't have ionic charges on their side chains, and polar acidic and polar basic. Here's the basic structure of an amino acid at pH 7. It has the same amino acid backbone. So you have a protonated amine and a carboxylate with a carbon in between. These are the two functional group forms that you'll see at pH 7. But you could also see this as an amine and this as a carboxylic acid. Attached to this carbon, which is called the alpha carbon, is an R group. R in chemistry is kind of like X in your math class. It means it's a variable group. So this R means these variable groups that are different on each amino acid. Notice that each of the amino acids has the same backbone group that I've highlighted here in blue. And they all have different side chains, which are highlighted in pink. Take some time now to please highlight these parts of the amino acids in different colors. Use highlighters or colored pencils to distinguish between the backbone groups that are the same between all amino acids and the side chains, which are different. Each amino acid will have the same types of functional groups, either amines or protonated amines, and carboxylic acids or carboxylates. I'd also like you to notice that each one of these amino acids has a name depending on what the side chain is. And each name has a shorthand version, a one-letter abbreviation or a three-letter abbreviation. You'll want to get familiar with the structures of each of these amino acids, their names, and their different abbreviations in the one-letter abbreviation or the three-letter abbreviation. But you should always have a chart ready to help you so that you don't have to memorize these things. Now remember what this type of structure means. This is a skeletal structure, and it means that there's a carbon at each of these spots where the lines intersect. So this carbon is the alpha carbon, and this carbon is the carbonyl carbon. Notice that the alpha carbon is a carbon that has four different groups attached, and so it's a chiral center. I can indicate that the alpha carbon is a chiral center by adding an asterisk to either of these structures. Take some time now to label the chiral alpha carbons in all of the amino acids. Only label it chiral though with an asterisk if it's actually chiral. Pause the video now and try to label all of these chiral carbons. Okay, now I'm going to label all of the chiral alpha carbons with an asterisk. All of the amino acids except for one have a chiral alpha carbon. Glycine is not chiral. Notice that its side chain is a hydrogen. And there's actually another hydrogen here, it's just not drawn. So here I'll draw it. Because this carbon is attached to two different hydrogens, and both of those hydrogens are the same as one another, this carbon is not chiral because it is not attached to four groups that are different. So glycine is the only amino acid that is not chiral. There are two amino acids that have an additional chiral center, or they have two chiral centers instead of just one. Pause the video now and see if you can find the two amino acids that have a second chiral center. Isoleucine has a chiral center here, and threonine has a chiral center here. Isoleucine and threonine are the only two amino acids that have two chiral centers. All amino acids except for glycine are chiral, and isoleucine and threonine have two chiral centers. Now I'd like to focus on the different categories of amino acids. And we'll start with the nonpolar amino acids. Here's a closer look at the nonpolar ones. These amino acids are classified as nonpolar because the side chains of these amino acids are nonpolar or mostly nonpolar. Take a moment now to classify each of the side chains into a functional group, family, or class. Most of the side chains on these nonpolar amino acids have nonpolar side chains, and most of them actually fit into the category of hydrocarbons. There are only 3 of these that can actually be listed as a functional group. Methionine has a functional group called a thioether. Tryptophan has an aromatic group And this is actually a special type of aromatic group that also includes an amine. This is called an indole, and it's a special aromatic amine that is not basic like other amines usually are. And phenylalanine is an aromatic functional group. Most of these side chains can be classified as hydrocarbons. There's one thioether, one indole, and one aromatic side chain. These amino acids are classified as nonpolar because their side chains are mostly nonpolar. Nonpolar amino acids are hydrophobic. They have hydrophobic side chains because they don't interact well with water. Remember that the word hydrophobic means that it's not water soluble, and the word hydrophilic means it is water soluble. All right, pause the video now and try to answer the next few questions on your own. The polar neutral amino acids include these families of functional groups. There are alcohols, phenols, thiols, and amides. These functional groups are not acidic or basic. They're called neutral because these functional groups do not participate in acid-base reactions and will almost never have an ionic charge on them. On the other hand, the polar acidic and polar basic amino acids have functional groups that are acidic and basic. The acidic functional group, carboxylic acid, is present in both of the amino acids that are classified as acidic. They're classified as acidic because their side chain functional group is acidic. At neutral pH, these things will actually exist as the conjugate base version of this functional group, a carboxylate. Look at the first page of your activity to see another representation of these two amino acids where the carboxylic acid is written as the carboxylate. The carboxylate is the form at neutral pH, or pH 7. Polar basic amino acids contain amines, or special aromatic amine functional groups. You should know that amines are weak bases, and these are characterized as basic amino acids because they have basic side chain functional groups. Basic side chain functional groups will exist as their conjugate acid version at pH 7. If you want to see what these look like at pH 7, please turn to the first page of your activity and examine the structures that are shown there. On the first page of your activity, these are represented as their conjugate acid form. Pause the video now and try to complete this activity, including the check for understanding. When you get to the last question, question number 3 on your check for understanding, unpause the video and we can go through it together. This is number 3 on your check for understanding, and this is a challenge problem, and it includes the review from a previous activity. Click the link shown right now to review the activity that we've seen before. The first time we did a problem like this, we were using alanine. Alanine is an amino acid that only has 2 functional groups. Now we're going to apply the same type of strategy to tackle histidine. Histidine has 3 functional groups. This problem tells you that the pKa's of histidine are 1.82 for carboxylic acid, 6.0 for the protonated aromatic amine, and 9.17 for the protonated amine. This is the regular amine that's part of the backbone. The 2 versions of the histidine side chain are shown on your activity on the first page and the third page. Draw the major form of histidine at the following pHs, and there are a bunch of pHs listed. You'll follow the same sort of strategy that we followed for alanine. First, draw out the pH scale and map the pKa's onto the scale. Something like this. Here's the pH scale, and here are the pKa's. Notice that I've labeled each of the pKa's on the scale, and I've labeled it with the functional group that it corresponds to. Now, I'll want to draw the structure and basically label these different pKa's to the different functional groups on the structure. I like to start at the most acidic pH, at pH 0. At pH 0, all of the functional groups will exist in their conjugate acid form. So, the amine on the backbone will exist in the protonated amine form, the conjugate acid form. The carboxylic acid on the backbone will exist in its conjugate acid form, which is the carboxylic acid. And the side chain will exist in its conjugate acid form. To see the conjugate acid form of this side chain, look at the first table on your activity. In each of these functional groups, I've highlighted the acidic hydrogen. When the amine converts to its conjugate base form, it will lose one of these hydrogens that I've highlighted in green. When the carboxylic acid converts to its conjugate base form, it will lose this acidic hydrogen on the carboxylic acid. And when the protonated side chain converts to its conjugate base form, it will lose this hydrogen. I know it's this hydrogen because this is the nitrogen that's positive, and so this is the protonated amine, the conjugate acid form of an amine. I've labeled the pKa's of each of these functional groups on the structure. Now ask yourself, which is the most acidic functional group? It should be the one with the lowest value pKa, or the smallest pKa value. The smaller the pKa value, the stronger the acid. And the carboxylic acid is the strongest acid functional group here. You can also tell this based on the map that you've made. This is the acidic side of the spectrum, and here is the most acidic pKa. It's the one that's closest to 0. This is the form of histidine that would exist down here at pH 0. Now I'm going to imagine if I start increasing the pH, what would happen to the amino acid? It would exist in the form that's shown here all the way up until pH 1.82. And as I approach this pH, there would be a change that happened, and it would be a change in the carboxylic acid. At pHs below the pKa of a functional group, that functional group will exist as its conjugate acid. And at pHs above the pKa, that functional group will exist as the conjugate base. In this case, it's the difference between the carboxylic acid and the carboxylate. If I wanted to know what the structure would look like above this pH, I would just redraw the structure where the only change would be this functional group. So I'll do that now. Notice that I've removed this hydrogen ion and I've left a negative charge behind. This is the structure that would predominate in solution below pH 1.82. Above pH 1.82, in this area here, this is the major structure that would exist. And at pH 1.82, you would have 50% of each of these structures. Okay, now let's think about what would happen as we approach the second pKa, as we increase the pH towards 6. There would be another change that would start happening. And it would be a change at the side chain, because the side chain has the next most acidic functional group. So the next change that would happen would be to this functional group. If I want to see what histidine looks like up here, I would just redraw the structure where I changed just this functional group. At any pHs below 6, I would have the conjugate acid as it's drawn here. But at pHs above 6, I would have the conjugate base form of this functional group, where I lost a proton. This is what it would look like. So any pHs between 6 and 9.17, this will be my predominant form of the amino acid histidine. In this zone here, between 6 and 1.82, this would be my dominant form. And at pH 6, I would have 50% of each of these two structures. What would happen as I approached pH 9.17 and even past pH 9.17? The change that would happen here is associated with the protonated amine that's part of the backbone. This one. This is the last acidic hydrogen left on the structure. And as I increase the pH, this functional group will change into its conjugate base form, the amine. This is what this structure would look like. At any pHs between these two pKa's here, this would be my dominant structure. But at pHs above 9.17, I'm now above the pKa of all of the functional groups. And so this is the most basic form of this amino acid. Now that I've drawn a map of all of the possible structures, it's easy for me to identify which structures exist at different pHs. Now try to complete this problem by answering the question. Check the key to make sure you're correct. Thanks for watching this video about the characteristics of amino acids. Make sure you take the time to review the necessary information so that this material is clear. If you have any questions, feel free to post them in the comments, and please let me know if this video is helpful.